Okay, so what organism can survive 3 million rads of radiation? For context, that is like 3,000 times the lethal dose for you and me. Or how about thriving at a pH of 0.03, which is literally acidic enough to dissolve solid metal? Now, if you're picturing some bizarre alien life form, you aren't looking closely enough at our own planet. These are the extremophiles Deinococcus radiodurans and Picrophilus oshime. Radiodurans doesn't just survive massive radiation, its genome gets completely shattered into hundreds of pieces, and it possesses this rapid DNA repair mechanism that flawlessly reassembles its entire genetic code within hours. It's wild. As undergraduate biology students, you've likely encountered the term prokaryote, and maybe subconsciously linked it to the word primitive. Well, today we are going to completely shatter that illusion.
Welcome to this explainer. Our core narrative today is simple but profound. Prokaryotes are absolutely not primitive. They are highly evolved, ruthlessly efficient, dominant biological machines that literally run the biosphere. For 3.5 billion years, they have been adapting, surviving, and perfecting the molecular machinery that keeps everything else on Earth alive.
All right, let's dive into this. We're going to cover 1. Cellular and molecular architecture. 2. Drivers of genetic diversity. 3. Metabolic mastery and ecology. And 4. Human impacts and biotechnology.
Starting off with Section 1, Cellular and Molecular Architecture.
To really understand these organisms, we've got to zoom way in, down to the nanometer scale. We're looking at the molecular defenses and propulsions that make the prokaryotic cell such an unyielding fortress. A fundamental division in bacteria architecture is revealed by the Gram stain. Think about gram-positive bacteria. They have this thick, robust layer of peptidoglycan, which is a polymer of modified sugars cross-linked by short polypeptides. Because of that thick layer, they trap crystal violet dye and stain purple. But then compare that to the gram-negative architecture. They have a much thinner layer of peptidoglycan sandwiched under a highly complex outer membrane. And this outer membrane is packed with lipopolysaccharides, basically carbohydrates bonded to lipids. So why does that matter to us? Well, the lipid portion acts as a potent endotoxin that triggers a massive, sometimes fatal, immune overreaction in hosts like humans. More importantly for medicine, this outer membrane acts as a formidable shield. Its highly selective porin channels actively block the entry of many life-saving antibiotics, making gram-negative infections notoriously difficult to treat in clinical settings.
And this brilliantly illustrates the stepwise molecular evolution of one of nature's absolute coolest structures, the bacterial flagellum. A flagellum's motor spins at up to 100,000 revolutions per minute. But how does something that complex even evolve? Well, genomic analysis shows us that it wasn't built from scratch. Step 1, the cell already had an ancestral secretory system used to move proteins across the membrane. Step 2, proteins homologous to ion transport channels were added, forming the base of a rotary motor powered by a proton gradient across the cell membrane. Step 3, a pilus-like ancestral protein was modified to add the structural rod, the curved hook, and the long filament. Think about this mind-blowing statistic for a second. 19 of the 21 essential proteins in this motor are modified versions of proteins that initially did completely different jobs in the cell.
Which brings us to a really crucial evolutionary concept, exaptation. Exaptation is the process where structures originally adapted for one specific function take on entirely new functions through descent with modification. So the flagellum isn't a product of sudden, spontaneous engineering, it's a masterpiece of exaptation. Biology is the ultimate tinkerer, right? It repurposed existing secretory and transport proteins to create a high-speed rotary motor entirely out of spare parts.
Moving right along to Section 2, Drivers of Genetic Diversity.
So, this structural brilliance kind of begs a question, right? How do prokaryotes adapt and evolve so insanely fast? Especially since they reproduce asexually through binary fission, essentially just cloning themselves. 8 million. That is the approximate number of new genetic mutations generated by E. coli in a single human intestine every single day. While the probability of a mutation in any given E. coli gene is exceptionally rare, we're talking about 1 in 10 million per cell division, the sheer scale of their reproduction changes the math completely. When 20 billion new cells arise daily in your gut, they are essentially buying billions of microscopic evolutionary lottery tickets. The raw material for natural selection here is staggeringly immense.
Now, what's really interesting about this next bit of data is that we can see this happening in real time. Take Richard Lenski's famous long-term evolution experiment. Lenski's team tracked E. coli populations founded by a single cell in a highly challenging, low-glucose environment.
When we track the timeline, by year 2, at generation 5000, the population's relative fitness and growth rate had skyrocketed as they rapidly adapted to that lack of glucose. And by day 3000, at generation 20,000, they were still accumulating beneficial mutations, fine-tuning their metabolic pathways to squeeze every last ounce of energy from their environment. This empirical data proves that rapid reproduction, when combined with mutation, leads to incredibly fast, observable evolutionary adaptation.
But hey, mutation isn't their only trick. Prokaryotes also engage in horizontal gene transfer, recombining DNA from entirely different cells, and sometimes even different species. There are 3 main molecular mechanisms for this. First is transformation, where a cell actively scavenges and takes up foreign DNA fragments directly from its surrounding environment. Second is transduction, where bacteriophages, which are viruses that infect bacteria, accidentally package host DNA during replication and carry those host genes from one cell to another. And third, conjugation. This is where a donor cell uses sex pili to attach to a recipient, reeling it in to form a mating bridge to transfer DNA. This process is driven by a specific piece of DNA called the F factor, which is often located on a highly mobile plasmid. This genetic swapping is exactly how multi-drug antibiotic resistance genes spread like wildfire through bacterial populations.
Okay, Section 3, Metabolic Mastery and Ecology.
So, having covered their genetics, let's examine their ecology. The crucial point here is that prokaryotes vastly outperform us, eukaryotes, when it comes to metabolic versatility. Pretty much every nutritional mode you can think of exists here. Take photoautotrophs, like cyanobacteria using light and CO2, but then consider the chemoautotrophs. They don't even need light. They are literally extracting energy from inorganic chemicals, like deep-sea hydrothermal vents spewing hydrogen sulfide or ammonia to fix carbon. It's like alien chemistry happening right here on Earth in total darkness. Then we have photoheterotrophs, unique aquatic prokaryotes that use light for energy but must consume organic compounds for carbon. And finally, chemoheterotrophs, which consume organic compounds for both energy and carbon, which is pretty much what we do.
But what happens when a single cell just can't do it all? Well, they cooperate. In the filamentous Cyanobacterium anabina, individual cells face a major biochemical conflict. The oxygen produced during photosynthesis actively degrades nitrogenase, which is the exact enzyme they need for nitrogen fixation. So what's their solution? Metabolic cooperation. Most cells in the chain carry on photosynthesizing, but a few differentiate into specialized cells called heterocysts. These heterocysts develop significantly thickened cell walls to block out atmospheric oxygen, totally protecting that sensitive nitrogenase enzyme. This allows them to fix nitrogen and share it with their photosynthetic neighbors. It's a brilliant, microscopic division of labor economy.
And finally, Section 4, Human Impacts and Biotechnology. Let's move to and see how this builds upon our earlier molecular discussion of cell walls, as we shift to the intersection of prokaryotic biology and human civilization. Pathogenic bacteria cause disease primarily through toxins, and these fall into 2 distinct molecular categories. Exotoxins are dangerous proteins secreted actively by living bacteria. Vibrio cholerae, for example, secretes an exotoxin that binds to our intestinal cells, activating a signaling cascade that forces them to release chloride ions. This draws massive amounts of water out of the body, causing severe, life-threatening dehydration. Endotoxins, on the other hand, are the lipopolysaccharide components of the gram-negative outer membrane we talked about earlier. These aren't secreted. They are released only when the bacteria die and their cell walls break apart, causing dangerous systemic immune reactions like severe fever and septic shock, which you see in salmonella infections.
So, to fight these pathogens, we are continuously searching for new antibiotics, often sourcing them directly from soil bacteria. Let's analyze some experimental data testing a novel soil-derived antibiotic called Teixobactin against MRSA, a deadly, drug-resistant strain of staph. Teixobactin is unique because it binds to highly conserved lipid precursor molecules right in the bacterial cell wall, making it exceptionally hard for the bacteria to mutate resistance. Keep in mind, this data is measured on a log scale, so a decrease of 1.0 means a 10-fold drop in bacterial colonies. The control group sits high at around a log of 9.0. Vancomycin, which is a standard heavy-duty clinical antibiotic, drops the MRSA colonies to around 5.0. But look at the results for Teixobactin at the exact same dose. It drives a much steeper, more significant reduction, plunging MRSA colony logs down into the 3s and 4s. That represents a massive, exponential clearance of the infection, and it perfectly highlights the biochemical firepower we can harvest from the prokaryotic world.
But honestly, perhaps our greatest harvest from these organisms is CRISPR-Cas9. This is literally a bacterial immune system.
Prokaryotes use short DNA repeats and CRISPR-associated or Cas proteins alongside guide RNA to recognize and chop up the DNA of invading viruses. Human researchers recognize this massive exaptation opportunity and repurpose this molecular machinery. Today, we use CRISPR-Cas9 to guide precise cuts in our own genomes. The guide RNA locates the target sequence, and the Cas9 protein acts as a pair of molecular scissors. This allows us to edit genes, fix mutations, and potentially cure genetic diseases. We have literally taken the ancient molecular defenses of bacteria and turned them into the ultimate scalpel of modern genetic engineering.
Which leaves us with a pretty staggering thought to conclude our explainer today. Prokaryotes have been the undisputed masters of the biosphere for 3.5 billion years, constantly iterating, mutating, and perfecting the molecules of life. Now that we, a relatively young eukaryotic species, have begun to master and wield their ancient molecular tools, like the CRISPR system, to edit our own genetic code, how is this going to alter the very future of human evolution? It's a massive question that bridges their microscopic past with our macroscopic destiny. And honestly, it is entirely up to your generation of biologists to answer it.